Okay, let's jump right into this source material and figure out exactly why DNA, and not its super close cousin, RNA, ended up becoming the dominant genetic material in most living organisms. We're setting up the ultimate biological showdown today.
So, why is DNA the master of our genetic code? I mean, we always hear about our DNA blueprint, right? But RNA kind of seems to take a back seat. To really answer this, we need to treat the role of genetic material like a highly competitive job opening. Let's see exactly how our 2' molecular applicants measure up.
Here's our roadmap for the showdown. First up, we'll outline the genetic job description. Then, we'll compare replication and gene expression. Third is the chemical stability showdown. After that, mutation rates and evolution. And finally, the ultimate verdict: storage versus transmission.
All right, moving into Section 1, the genetic job description. We're looking at the 4 absolute rules a molecule has to pass to be the master molecule.
To even get an interview for this job, a molecule has to pass a rigid 4-point checklist. First off, it's got to be able to generate a replica of itself. Second, it needs to be chemically and structurally stable. No falling apart on the job. Third, it has to provide scope for slow changes, mutations, to allow for evolution over time. And fourth, it must be able to express itself as Mendelian characters, which basically means it can actually dictate physical and biological traits. So bottom line, any successful genetic material absolutely must hit these 4 precise criteria to properly manage the blueprint of life.
Let's step into the ring for round 1. Section 2, replication and gene expression, focusing on duplication and coding.
Now, for that first requirement, generating a replica, both of our applicants actually pass with flying colors. Thanks to the really neat rules of base pairing and complementarity, both DNA and RNA have this built-in ability to direct their own duplication, which is actually a huge deal because other vital molecules in our bodies, like proteins, they completely fail this first test. They can't copy themselves at all.
But what happens when we look at the 4th rule, expressing traits? This is where RNA actually takes an early, massive lead. See, RNA can directly code for the synthesis of proteins. It easily and efficiently expresses characters all on its own. DNA, on the other hand, it's completely dependent on RNA for protein synthesis. DNA literally has to hire RNA to do the heavy lifting of actually expressing those traits out in the cell.
Which brings up a really fascinating point from the text. The protein synthesizing machinery has evolved around RNA. This brilliantly illustrates just how fundamental RNA is to the actual creation of the proteins that build our bodies. Honestly, if we were just judging on who does the active day-to-day work, RNA looks like the clear winner right now.
But wait, there's a catch. Let's head into Section 3, the chemical stability showdown. This is round 2, the stability test, and it's where we find the most critical difference between our 2 contenders. Before we get into the specific differences, just take a second to visualize a polynucleotide chain backbone. This microscopic structure is exactly where we'll find the chemical quirks that define both DNA and RNA. It all comes down to the tiny details right here on this chain. I want to introduce a really crucial term from the source here, labile. It essentially means something that is easily degradable, highly reactive, and super prone to chemical change. Keep this definition locked in your mind, because being labile is RNA's absolute biggest weakness in this job interview.
Here is the structural smoking gun. In RNA, every single nucleotide residue has an additional 2' OH group. Just that one specific extra oxygen-heavy group makes RNA highly reactive, easily degradable, and catalytic. In other words, it makes RNA highly labile. Now, DNA structure is missing that specific group, and because of that, DNA is chemically far less reactive. Think of it like this: that extra group makes RNA highly social and reactive, while DNA is more of an introvert. It's less reactive, and therefore a much safer vault for storing our vital information.
And the chemical differences don't stop there. Adding to DNA's impressive structural defense is its choice of nitrogenous bases. DNA uses thymine, which naturally gives a boost of chemical stability to the whole structure. RNA, though, uses uracil in place of thymine. It seems like a minor molecular swap, but missing out on that thymine makes RNA significantly less stable over time. You can actually see this physical resilience in action. Think back to Griffith's famous transforming principle experiment. When he killed bacteria with extreme heat, their genetic materials wasn't destroyed. Why? Because DNA is so structurally rock solid that even when you heat it up enough to completely separate its 2 complementary strands, those strands will literally just reunite and snap back together once things cool down.
RNA just cannot compete with that level of stability.
Okay, moving to Section 4, Mutation Rates and Evolution. Welcome to Round 3, the mutation race, where we look at the need for slow evolutionary changes.
So, both DNA and RNA can mutate, which is obviously essential for evolution, but their differing stability completely dictates the pace. DNA mutations happen at a much slower, controlled rate, precisely because of its robust chemical stability. RNA mutations, however, happen at a blazing fast rate because the molecule is just so reactive and unstable. While RNA can mutate, its structural instability means it mutates way too quickly to be a reliable long-term blueprint for complex organisms that need to stick around for decades.
This chemical reality perfectly explains the behavior of viruses. Viruses that rely on an RNA genome have a shorter lifespan, they mutate incredibly fast, and as a result, they evolve at a blistering pace. So the key takeaway here is that this rapid mutation rate, driven entirely by RNA's inherent chemical instability, is exactly why RNA viruses are constantly shape-shifting and are so notoriously hard to pin down.
All right, let's dive into our final section, Section 5, Storage versus Transmission. It is time to finally deliver the verdict on our genetic material.
When we lay out all the evidence, we see something pretty amazing. Look at chemical stability. DNA is highly stable thanks to deoxyribose and thymine, while RNA is reactive and labile, thanks to its 2'OH group and uracil. Mutation rate? DNA is slow, RNA is fast. Because of these hard facts, rather than picking one absolute winner, biology did something brilliant. It issued a division of labor between the two applicants based entirely on their unique chemical strengths.
Biology assigned them very clear roles. DNA, being chemically and structurally much more stable, is the ultimate choice for the storage of genetic information. RNA, on the other hand, is perfectly suited for the transmission of that information. So DNA gets the job as the secure, long-lasting, unchangeable vault for our genetic code. Meanwhile, the highly active catalytic RNA takes on the dynamic, everyday job of transmitting that code out of the vault to actually build and run the organism.
Which leaves us with a truly fascinating mystery to ponder. If DNA is the ultimate storage vault today, but our entire protein-building machinery originally evolved around the highly reactive RNA, did life originally begin in an RNA world? I mean, if DNA is essentially just a chemically modified evolutionary upgrade designed for better stability, does that mean the unstable, fast-moving RNA was actually the very first spark of life? Think about that the next time you hear someone talking about their DNA. Thanks for joining me for this explainer, and keep questioning the science behind our existence.